we get experimental science. We're curious, non-judgmental.
There we go, that's much cleaner of color. All right, just to give y'all an idea, the actual piece is 2 centimeters in length, 1 centimeter in width, and then maybe 2 millimeters in width. I think you're on the right trail with this. The eyes, eye type in particular to the range of the size of the head. The antennas are also a little bit like fly-like, as it were. Any guesses as to what that is, Professor Plodicus? Right here, this little nub, is an antenna. That antenna is very, very much a fly antenna. So it's essentially this nub, and then it has this hair sticking out. And that's your standard fly antenna. Also here, Tommy, is a haltere. Look at that. And then here is the other one, actually. There's the other one. So you might be wondering, what the heck are these halteres? That is a mark of a true fly. So, Sam, too small to be a housefly, if you go by modern day houseflies. This is kind of like what ours looks here in terms of relative size to the body. So here and here are 2 halteres. Here and here. So again, those are the halteres of our insects. This is 30 million years old, preserved in a piece of ancient resin. I think it's really beautiful. The antennal structure is another segment of what we're able to be seeing here. So I think this is a neat way of looking at it too, is that we've got these halteres on these flies, and then this is going to be showing like takeoff flight of them. And again, if the idea and the hypothesis is that these halteres are meant to assist with flight, we should be able to see them. So take a close look. Right here is where that haltere is going to be. And it's actually going to be wiggling up and down. And again, look closely right here, y'all. Look at how cool that is. There's still actually controversy on what these halteres are used for, whether or not it is for balance, what part of flight it's for, right? Because it is moving along with it, but there's no way that that flapping is going to lift anything. The reason, Cliff, I'm pointing this out, so right here, for again, anyone who's coming in, we are guessing together as a group and try to identify what's here in the specimen. And so looking at those specimens, we did see the haltere. And the haltere, I think, looks really apparent, really easy to find, and it just makes me really, really excited to see something like this, chat. Like, this piece of biology is maintained in today's animals as well, is the use of those halteres in flight, which again, I think is really, really cool that something like this still exists today in our animals. And actually, let's do a quick comparison. How about, chat? I have, of course, crane fly specimens that are just like from Jurassic Park that we can take a look at together and have a little bit of that comparison of those halteres. So if we zoom on out, there is our crane fly. Let's see if this one's halteres are how visible they are. Of course, y'all, this one's the difficult one to find. We've got a bunch of bubbles, but they'll be located right around here. So you see there's a really like big bubble that like kind of blew up the internal anatomy. And so we have right here is that haltere coming out. So the sample that we have here, again, I'm leaning towards this being a fly. Could it be a forward fly? Hump antennae would fit. I think so, Cliff, and it's also a similar size. So let's go ahead and bring up forward fly image. Forward flies for our leafcutter friends are terrifying. They are parasitic. Ooh, Cliff, you got it. I think this is it, Cliff. So there's a secondary structure here that I think Cliff Alster-McLean has picked up on. Cliff, is it this right here? These little nubs? Nub 1 and nub 2? So those two are, I think, what Cliff is picking up here on this forward fly. So not only do you have those antennae, but you have that additional structure coming out as well. And so those two together, I think we're leaning very close on this being some flavor of forward fly. These animals infect the brains of ants. So what they'll do is they'll inject an embryo and venom components into the head of a leaf carrier ant, let's say. And as the ant is carrying the sample, like a leaf, back to the colony, this injection process happens. And when that happens, there's a little larva that's now growing inside of the brain of the ant. And then the ant's brain gets eaten, and from the head erupts this forward fly. It does bring a question, y'all. This is a good idea, right? Because what might happen if there had too many of these forward flies are released? right? If there's too many of the forward flies released, then what do we do? This does bring up the question of if it's a worthwhile endeavor to use them as biocontrol, right? Because what if they started going after something that, I don't know, would be less than optimal, right? That they might start taking out something different. And they're also releasing forward flies that, yes, specialize on fire ants, but there's nothing to suggest that they couldn't specialize on something else afterward. And then the hope is that these forward flies will help calm that population spread. I think we're having fun. And yeah, this is a really cool specimen. I don't think that we've ever got one of these samples before. Mini parasites, essentially. They will inject their eggs into the heads of primarily ants, is what it's been studied, although there's forward flies that prey on other exoskeleton-based animals. So there is in fact an entire caste of leafcutter ant dedicated to fighting these off. So what you can imagine is there's a leafcutter ant that carries a leaf to and from the colony and the site where they go and chop up the leaves, right? As they're carrying the leaves, they are unable to fight off predators. And so there's a caste of leafcutter ant that rides on the leaf and fights off forward flies that may come and attack the carrier. So there's the carrier caste, and on top of that leaf that's being carried is the rider caste. I believe, hold on chat, I might have a picture. So you can kind of see it here. There is your leaf carrier, and then this right little ant here is your leaf rider. So those two are acting in unison. This ant is carrying this leaf, and the rider is hanging out. If the forward fly were to come near the head of this carrier, this rider would fight it off. But what we did, we took other ant species and glued them to a stick. There was a recently expired ant species, carpenter ants, and then we put them on a stick and we fluttered around the leaves that were being carried. Is that really janky? Yes. Is it eyebrow-raisingly janky? Yeah, definitely eyebrow-raisingly janky. But, chat, it did elicit the behavior. So all of a sudden, then the leaves that were starting to be carried had these riders on them. Now, the one big catch, y'all, is it did seem like we had to have a certain threshold of ant numbers. So the experiment didn't work if you had very, very few ants to start with. You needed a pretty large colony in terms of size, and then we started to get more and more of these events happening. But again, there is a particular threshold that we've got. And that threshold we've noticed be the case in other experiments as well. For example, you will not get queen ants, like new queens or males hatching from a colony unless your ant colony is rather large. With leafcutters, you need about 1 million ants before you'll start hatching reproductives. Why is that? Usually these density questions connect with some kind of, like, there's almost an inherent knowledge of the colony that's like, hey, we can feel that this particular density is how much we need in order to hit these additional goals almost for us to be able to increase our colony numbers to the point where we need to be better, and we need to be, like, we're at a point where we can have soldiers and these more energetically costly castes. I wonder if that's something that's going on here too. But yeah, we have here this forward fly, and again, the forward fly is being what the rider's trying to fight off. Forward fly injects that embryo into the head of the carrier. The first thing to go is the muscle tissue. And now you might be like, Balint, why is it gonna be the muscle tissue? Why not eat the brain? The brain is juicy. The brain is delicious. I saw the Indiana Jones movies. It's a delicacy, right? Somehow these parasites have a threshold and they know what they need to eat first. And so here, you can look at this image. This is a leafcutter ant head that we chopped open, and there is tissue back here. So we actually marked all of the musculature. And so we only got, what you see here as in terms of the image. But that's all muscle tissue. The brain isn't even visible here, but it would probably be like, this size. Like, in terms of the overall mass of what's in the head, the brain is nothing. Majority is muscle tissue hooked up to those mandibles that allows them to chew through the leaves. They need really, really powerful teeth and powerful ability to chew to be able to get what they need to do in terms of cutting those leaves. And so this image in red marks you cells. In particular here we have muscle cells and we have neurons as well. Actually, so Clayman Dev has discovered something very important to note, I think, Clay. The original sequencing experiment on ant brains was not actually on brains at all, Clay. What they did, the scientists cut the heads of soldiers and nurses off and put those into the sequencer. Meaning, Clay, that they weren't just doing one tissue, right? They weren't just dissecting out the brain. They were getting the entire head, right? And the entire head, for some of these casts, like the leafcutter or the soldier, has a ton of muscle, and the nurses have very, very little musculature. And so that led to this false naming system of brawny and brainy. And that's what they were looking at, those differences. So there was the brawny, which was the soldier, like you're saying, Clay, the meatheads, and then the brainy, which were the nurses, which didn't have them. But the problem is that the experiment was done wrong. They included the musculature in the sequencing. And because they did that, it biased the sample. If you remove the brain and do the sequencing there, actually dissect out the brain. So instead of having something like this, where you're looking at all the musculature, just remove the insect brain by itself and sequence that. So you'll have something like this. So this is the brain of one of these soldiers. You can see a lot of detailing here. Everything from the optic lobe, which is the visual processing center, to the center part of the brain. You're not going to get that muscle tissue, and therefore that whole conclusion of brawny versus brainy isn't going to happen, because it's not something important anymore. Does that make sense, Clay?